All right, thank you very much. So, for a non-mathematician, this is quite a daunting place to give a talk in front of an audience like you. In fact, when I was 15, I had to choose my A-level subjects at school, and my father went to the parents' evening and came home completely depressed because he had asked the math teacher, "What do you think Piet should do?" And he said, "Well, not math, maybe French and something arty." So, so that's been haunting me my whole career. So this is the pinnacle of my career. I've been working towards this moment, and I may send him a postcard tonight from Cambridge and say, "When was the last time you gave a talk at the Isaac Newton Institute, Mr. Knoll?" Anyway, because I'm a non-mathematician, I'm also going to be slightly controversial and try to entertain you the next 20 minutes or so with the idea that when we talk about uncertainty in cardiac modeling, very little actually comes from mathematical or statistical uncertainty, I'm afraid. And in my world, which is drug development, biology and the uncertainty in biology trumps everything. So I'm going to talk about pharmacokinetics and pharmacodynamics, and this slide is just to introduce that word. So this is a very old slide. This is actually the very first analysis I did at Pfizer when pharmacokinetic-Pharmacodynamics, or PKPD, was a very new topic. So this is probably almost 20 or 25 years old. And the very first data set I was given to analyze was a dog study where two compounds shown here were tested on the effect on the QT prolongation in the ECG. And certainly in these days that was a very, very hot topic because several compounds had been taken off the market because of that. So I did my magic and went to the biologists, and they were like, "This is absolutely amazing." So that's probably the best response I've ever had ever since on a piece of modeling. They said, "You know, this is incredible," because we always knew there was something different about these two compounds, and we always felt, you know, cisapride you had to really soak in before it did anything. So what you see here is a typical PKPD modeling approach where we have concentration on the x-axis. So this is the concentration in blood or at the site of action, and the effect in this case, the change in milliseconds in the QT interval for these two compounds. And so we can learn a lot of things from this. So first of all, we can kind of correlate the dose to concentration to the effect, which is very important. But also clearly these two compounds are doing something slightly different because the shape of the concentration effect relationship is very different. And only later I think that was rationalized by the fact that the blue compound is a very clean ion channel blocker, the hERG channel, and cisapride is a mixed ion channel blocker. So we can learn a lot from such PKPD experiments. Now the reason we do this in dog in pharmaceutical drug development is really because we want to translate this to humans. And that becomes important when you get a question from regulators like this. So here now you see a clinical data set, same as I showed you before, but now in man and with a different compound, and we have the concentration against the effect again. And to be honest, nothing happened. So the clinical team was pretty happy with this result because like most of these studies, the compound doesn't actually do much, which is good. However, then the regulators came back to say, well, yeah, but what if, you would have gone much higher than here? Because in the dog you did actually 12 times higher, so somewhere up to 500, and you saw an effect. So convince us, based on the data you have, that that is not going to happen in man. For example, if someone accidentally takes 2 tablets instead of 1. So that's actually very, very difficult to answer with. You can either do another experiment, a clinical trial, which is very expensive and wasteful, or you can use data like I showed you in the previous slide and make a compelling case that you know how to extrapolate that data from an animal to man. So that's the topic of this talk, because that sounds easy, but it's not. And the main reason is not because the mathematics is particularly difficult, it's because the biology in an animal may be very different from what it is in man. So the way we approach that is by generating data like this. So now you see data on another compound in another species, and we start off with lots of individual data on the left. So here we have PK, which is the pharmacokinetics, and PD, again, QT prolongation. And you see here a whole row of different doses in different animals. Someone asked earlier, I think it was you, said, do people ever think about doing population type modeling? Well, all the time. That's all we do in pharma. So to be honest, that is a very, very well established methodology as shown here and applied by regulators most of the time. So when we do that, we can convert the individual data, pool it all together and develop what we call a population PK/PD model, which now is a single model that captures all the inter and intra animal variability. So when we have that, we can do our magic and do the kind of translational PK/PD that we really need. And that's really because concentration is the kind of euro of pharmacology. Dose doesn't mean much. Dose is just what we give, but it's really the concentration, of course, that drives the pharmacology. So now you see again a plot, same as the ones before, where we have concentration on the x-axis, effect on QT on the y, but now we have three different systems. The one that I just talked about was the monkey data, which is the line shown here, with some uncertainty bands modeled because we had a population PK/PD model. In the middle you see in vitro data, so this is the Herc ion channel assay. And here you see several studies in man. So now you can see the beauty of doing PK/PD because we derived this common currency, the euro of pharmacology, and we can now all of a sudden compare in vitro with in vivo as well as different species, all in the same manner. So when we do that, we can infer some quite interesting observations and lessons. So in clinical studies, a 10 millisecond increase in the QT interval is seen as a red flag. Regulators would be very concerned if you would be reaching 10 or more than that. So what we can do now is we can kind of start to translate that clinically meaningful point to, for example, our in vitro assay. So we say, well, 10 milliseconds, the concentrations associated with that in the clinical studies range somewhere from here to here. And we can now read off our in vitro Herc assay how much % inhibition that is in that assay. And the answer is that's actually less than 5%. So that's virtually impossible to measure. There's virtually nobody still in the industry that can measure something less than IC5. You know, we typically use IC50, maybe IC20. Actually, this study kind of infers that we should be looking at something much, much less. We can also do the animal to man comparison in the same way. So we do the same trick. We kind of start at 10 milliseconds, see the concentration, look where that hits the animal curve, and now go back to this axis. And now you can see that actually a 10 millisecond change in man relates to a much less than 10 milliseconds change in this particular species. So that's the way we can kind of calibrate these assays and kind of do meaningful translation. And the typical idea that a millisecond in an animal relates to a millisecond in man is clearly flawed according to this. So the benefits of PK/PD is not just that we can learn more from it, is actually also a very efficient way of studying pharmacology, as shown in this study, which we did a few years ago in a consortium in Leiden, where we had a huge data set from pharmaceutical industry, and we compared the sensitivity of a classical statistical approach like ANCOVA versus PK/PD. And for a study design where we had 4 subjects in the study, we kind of looked at the curve where you see the ability to pick up a certain level of milliseconds increase versus the sensitivity. And as you can see in this particular assay with 4 animals, we reach 80% sensitivity for approximately 6 to 7 milliseconds, whereas with a classical statistical approach we can't even pick up 14 milliseconds. And remember from my previous slide, what we said is we need to pick up something less than 5 milliseconds. And when we go to 8, clearly the PKPD approach is very well powered, and a conventional statistical approach is still struggling to pick up a meaningful required level. So really, when you start to think about extrapolation in pharmacological sense, we're talking about extrapolation from one system to the other. So system can be from an in vitro assay to an animal, or from an animal to human, or from a healthy volunteer to a patient. And as I said, extrapolation here and the uncertainty that is linked to that really is mostly linked to the biology, because clearly a cell-based assay is not the same as a whole animal, and a rat may be quite different from man. So the more recent way that people have been starting to approach this is now kind of commonly referred to as quantitative systems pharmacology. It's been referred to earlier by Gary and Anna and others, where we really start to try to build models where we explicitly separate the properties of the drug, like binding affinity, which should be independent from whatever system you test it in, to the properties of the system, which could be the expression levels of the receptor or the turnover of the receptor. So when we start to apply such models to, for example, QT prolongation due to hERG-mediated ion channel blockade, we can build models that do pretty well in terms of being able to predict clinical studies, as you see here. So these are all lots of clinical studies, and a systems pharmacology model that actually predicts and simulates these studies without running them, entirely based on preclinical data. So when you look at that, particularly in the kind of lower ranges of relevance, so 0 to 10 or so milliseconds, this model does very well, which is why, and I wrote it in my abstract, my sense is the regulators are moving towards a point where such in silico simulations will become substitute for running actual clinical trials. I actually spoke to Janet Woodcock, head of CEDAR at FDA a few weeks ago, and she said, yeah, that will happen. I said, so when? She said, well, I can't tell you, but I think we're moving towards that. Now the big exception here is this one. So here you can see that clearly the model prediction, the blue line, doesn't kind of sit on the actual clinical data. It underpredicts the clinical effect quite significantly, which is bad in this case. Now this turns out to be the only pediatric data set in this whole cohort. So the rest were either older children or adults. So that's really interesting. So now we're starting to say, okay, apparently our model does pretty well for a certain population, but for a special population, in this case pediatrics, it doesn't. And now we can kind of start to play with our model and say, well, one assumption in the model is, for example, that the expression of the hERG channel is the same in very young children as it is in adults. And if we would kind of change that assumption, then all of a sudden we can kind of move our bad prediction, which was blue, to a pretty good prediction in orange. And that's simply based on the fact that if we assume that the hERG channel expression is twofold higher in pediatric children than it is in adults. So for several years I've been showing this slide without this. And I said, that's a really interesting hypothesis. Does anyone know whether young children express the hERG channel any differently from adults? Always silent. Nobody ever knew that. And then I found this paper in a Polish magazine. So this is the hERG channel. This is adult, and this is young children. And look, it's at least 2 times higher. This is RNA level expression, so maybe not quite the same as protein, but still pretty kind of in line with the model prediction. So back to my point, so all the uncertainty regarding this prediction was really related to the fact that we didn't know this. And really, if there's anything we need to do to decrease uncertainty, we need to invest in kind of getting data like that. So extrapolation, as I said earlier, is quite difficult. So this is not a cardiac example, but just to illustrate that point again. So this is data on another ion channel which was of interest as a target for treatment of pain, which is called the tryptamate receptor, which is a cold sensing receptor. And if you give modulators for that receptor to animals, you get a very pronounced lowering in the core body temperature, but it's very species dependent, as you can see here, where the smallest species, mouse, gives a very pronounced effect and the largest dog hardly any. So now what we can do is kind of build PK/PD models which includes allometric scaling, so models where some parameters are modified based on body weight, which is a very old methodology widely used in pharmacokinetics, not so much in pharmacodynamics. So we can build a simple model of temperature turnover, and we can say, well, the loss of temperature, we assume that is a rate constant that we can scale allometrically, where you see the mouse, the rat, the dog, and then man would be somewhat here. So if you do that, then you get model predictions like here, where you say, well, if we scale this to man, three things can happen. Either it's the worst case, man equals mouse, we get a big effect. Maybe we just take the mean of this, the median, it's somewhere between the three species. Or actually, if we're really optimistic, we're going to say, well, actually nothing will happen in man because it's based on allometric scaling. So the clinical study was actually run, and to cut a long story short, here you see the effect on body temperature, which is entirely circadian rhythm, and the compound actually did not do anything in man. Now in this case that was good news, of course, because lowering body temperature was a side effect, but you wonder how many failed clinical studies are due to this specific phenomenon, where we see fantastic effects in animal models and then nothing in man. And people always say, oh, you see, animal models are meaningless because they don't really represent human physiology. I think in many cases they are probably very good models, but the modelers are not particularly good at scaling them. So here's the challenge in cardiovascular. So I've listed from this recent paper just the kind of different heart rates, basal heart rates in different species and the different QTc values in milliseconds here. And the bottom line is, you see there's a huge range. In heart rate, going from 60 in man to 230 in guinea pig, and again also in the resting QTc, where very interestingly the animal most widely used is the outlier. So you wonder why we use that and not, I don't know, another one. And here's the challenge. So we need to translate this diversity of data to what we concern to be meaningful safety signals. So these are not signals in first-in-human studies where someone would fall over. So these are kind of very subtle effects which late in clinical development would be a very significant safety concern, not because at the individual level they are, but at the population level, a very small change in blood pressure could actually be a very kind of significant. And it's only something you will start to pick up when you study thousands of patients. So that's a real challenge, and that's really something that we're still to some extent struggling with, how to kind of be certain in the predictions of things like this based on data sets like that. So here's an example. Here now we switch from QT to heart rate. Again, you now are very familiar with PK/PD plots with concentration and effect, in this case heart rate, where now we see for the same compound the concentration effect in dog and the concentration effect in man. So here we have flipped the situation that I showed you at the beginning of my talk, where we said that man was more sensitive than dog. Remember, 10 milliseconds in man was only 5 or so in dog. Apparently for heart rate it's the opposite. So now all of a sudden man is less sensitive than man, as you can see in the slope here. And we did some literature survey and kind of found the relationship between the resting heart rate and the maximum heart rate that people have ever observed in a particular species. And you can see there's quite a complex allometric relationship there. So here's some more recent work moving up the kind of systems pharmacology scale in terms of how are we trying to develop more mechanistic models to help us understand how to scale preclinical data to clinical data. Now we're kind of starting to look at combinations of ligands. In this case, not necessarily drugs, but two key kind of players in cardiovascular regulation: noradrenaline, which acts at the adrenergic system, and vasopressin, which is one of the key hormones involved in homeostasis. So we can now build models along the lines of this, where we have ligand 1, which is noradrenaline in this case, binding to its receptor, forming a complex. Same for the other player, binding to a different receptor, forming its complex. But the idea is that if we do see interaction between these two players, which we do, in terms of the effect on contraction in small vessels, then these two things have to converge at some point to give the final effect. So we did lots of different studies, different combinations, alkylating one of the receptors to kind of knock it down. So we had lots and lots of data which really allowed us to fit a scheme like this. And the key thing here is, the most interesting bit is the estimation of the slope parameters. So this slope, which is the steepness of the relationship between this player and the intermediate, the same for the second player, and then the slope of the common converging pathway. So these curves are very steep. So what you see at the end is a very steep relationship. But until we did these studies, we didn't know whether that steepness came from this slope here, from that slope here, or from both. And in terms of the way the model behaves, it makes a huge difference. So anyway, we could do that, so we generated these kind of surfaces where we have the two agonists here and the response surface there. And to cut a long story short is that the steep slope that we observed appeared to be entirely driven by the steep slope at the end. And actually the intermediate concentration effect curves were very, very flat. And that has a huge impact on the behavior of the synergy that you see. So we now start to coin this outside-in systems pharmacology. So here we kind of generate lots of data. We don't infer a very detailed mechanistic model, but we kind of look at the fingerprints and then start to infer outcomes like this. Now the only way you can do this is if you have a system where you can do high throughput experimentation, because you need lots and lots of data to do that. Now we can also extrapolate this to man. So what I showed you was in rats. We can now kind of scale it to a human, and the simplest assumption would be man equals rats, and you can see that the prediction in blue doesn't really kind of sit on top of the data. We can then try various things, and it turned out that the most critical thing that we had to correct for was the difference in the receptor expression. And obviously we have two different receptors now that we need to take into account for. If we take that into account, similar to what we did with the hERG channel for pediatrics, we now get very good scaling to man. Okay, my last two slides is just some heads up for work that's going on now. If you want to hear more, Hugh sits in the back. She's the PhD student doing this work. So this is part of a large European consortium called TransQST, which is now the quantitative systems toxicology instead of pharmacology. And we're in the process of starting to analyze probably the largest data set that's ever been analyzed like this, in terms of cardiovascular hemodynamic data generated by 8 companies, and I think we now have 21 drugs in 4 species. And really what Hugh's going to do is to build a mechanistic model of hemodynamic regulation. So it looks a bit like this, where we have heart rate, stroke volume, cardiac output, mean arterial pressure, total peripheral resistance. And this model is obviously a system of ODEs, and it is something that we're planning to kind of use to kind of really understand how different compounds may affect this scheme in different places, because it makes quite a difference for a compound in terms of its safety liability where it actually impacts the hemodynamic system. So the first thing that you did is to look at what we call practical identifiability, is to, well, is such a model actually identifiable based on a data set that is practical to generate in a drug discovery setting? And the short answer to it, yes, it is. Obviously, the more you measure, the better, but even in a relatively sparse study where we only measure heart rates and stroke volume, this model appears to be identifiable. And if you want to hear and see more of it, you should go to the PAGE meeting next week, where she will be presenting this work in more detail. Okay, so I'll call this Back to the Future. I'm sure many of you have seen this slide where someone just wants to show the model of all models, and look at how big this is, you can't even see it. Anyway, my point is, so this is a model of hemodynamic regulation published by Guyton et al. In 1972. It's very interesting that we're now kind of almost back to the future, and we're using a much smaller model than this, but still mechanistic, and really start to now kind of think about, okay, possibly we can utilize models like this, maybe more constrained, to really help us understanding pharmacology and scaling it. So my very last slide is this one. So thanks to my colleague Mark Holbrook for providing this. So he did an overview, and this is a slide that shows you all the 65 drugs that have been withdrawn from the market due to something cardiovascular. And I thought it was quite interesting to show you that for several reasons. The first one is that the last time a drug was actually withdrawn from the market because of cardiovascular was almost 10 years ago. So I don't know what that means, but it seems to tell me that we're actually pretty good at this stuff now. I think we always beat ourselves up and say, oh, this whole issue. I think we've become pretty good at this, unless, of course, we're so cautious now that all this shows is that our method is very sensitive but not very specific, and there may be a lot of false positives there that we have thrown away. Obviously this doesn't show compounds that never reached the market and were kind of stopped during clinical development. The second point here is that it appears to me that all the emphasis in meetings, well, this meeting actually wasn't the case, but most meetings I go to, all the emphasis is on on ion channels, on arrhythmia, on QT, on multiple ion channels. Actually, that is a relatively small part of this pie. Well, small, a third. 2/3 is actually different cardiovascular events, like blood pressure, heart rate, cardiac injury. And it seems to me that maybe we should be shifting our emphasis slightly towards that topic as well. With that, I would like to thank you, and back to you, Gary. Everybody wants a drink.
Yeah.
Well, sorry to delay the drinks. Just so here you mentioned that in the last 10 years there's no drugs that was removed, but pro-arrhythmia there are 23 drugs, right?
Yeah, but that's since that was, well, obviously the majority were kind of withdrawn in the mid 90s when terfenadine and cisaprides lighted up as hERG channel blockers, and all of a sudden, of course, there was a whole bolus of compounds that were taken off the market.
It's just for clarification, for the pro-arrhythmia, is this meaning tertiary 1, or is this just any type of arrhythmia? What's the classification? How dangerous are these drugs?
Oh.
Well, that just depends on.
So I've been told that when you go and have your drink, and you'll have maybe I don't know if there's any snacks, but say there is, Your QT will go up by more than 10 milliseconds simply by just eating. I've never heard anyone kind of being overly concerned about that. So how dangerous is a 10 millisecond increase? I don't think it's particularly dangerous. On a population level, of course, there is a risk there that is very hard to justify. So that's why I think it's been implemented.
Yeah, thank you for the talk. I was wondering if you have that relation that the heart rate in men has a different slope as compared to the concentration as related to the dog, then the man is saved by the bell because of Bazet's formula, because that will underestimate the amount of influence on the QT interval. So why is then that industry is still going for the QTC and not for the QT? Never thought about that.
Is Nick McMahon still in the audience, or has he left? I don't know. I don't think I can answer that kind of straight away. It's either a brilliant thought or there's a very simple answer to it. I don't know.
But you're using Bazet's.
Oh, it's always the corrected QT that's used. Yeah, never uncorrected.
That's to underestimate the problem in the higher heart rate.
Yeah.
So, but okay.
Pradeep Sharma from AstraZeneca Safety Modeling Group. Thanks a lot for a very nice presentation. In one of your slides, you showed that some of the CV parameters, there's difference between human and the dog, which is QTC heart rate. So how did you came to that scaling? Is that scaling based on one set of drug, or it's based on some particular group of drugs which actually showed the similar pattern of difference in those parameters between dog and the man?
So you mean the table with all the different species?
Yes.
No, that was not. That was just basal. That was just basal heart rate, basal QT.
That's just the baseline response.
That's just baseline. That wasn't drug induced. No, that's just baseline.
Okay. So would that mean we can't use that scaling when we are doing first in man prediction?
No, I think the point about that slide is that clearly if you have a baseline that sits here and a baseline that sits there, then the same delta may be something quite different. So the same absolute delta on a very low baseline is clearly a much higher relative effect than the same absolute delta on a much higher baseline. So I think that's the question, whereas we typically still often just assume that, well, a 10 millisecond change in one species is 10 milliseconds in the other. That's still how, to a large extent, a lot of the preclinical data are being interpreted.
Okay, so if we are going to have, say, control or vehicle in PKPD modeling, would we not be correcting that baseline difference?
Yeah, so any change in baseline would be corrected,
Yeah.
but you would still end up with a conclusion that my drug causes a 10 millisecond change at this particular concentration. Now that 10 milliseconds may be something completely different in another species, because it doesn't translate one-to-one biology, and it depends on many factors.
Okay, well, we
Just
can do this over the wine reception. So, thanks to all the speakers today. It's been fascinating talks. Brilliant from my point of view. We've now got a wine reception, and don't miss the